Hi, I am Cabin Savage. I'm doing A-level biology, and in this video we're going to be going over things that in photosynthesis that are likely to come up in the test tomorrow in the January module. Basically, this one is going to be the effect on the levels of GP, RU, BP and TP on when the carbon dioxide, light and temperature levels change. So we've seen that changes in CO2, light intensity and temperature, obviously they all alter the rate of photosynthesis. So we'll start off with light intensity. Well, light intensity gives a measure of how much energy is associated with the light. Light from a source spreads out, and so if the distance from the source is doubled, the light intensity is quartered. It follows the inverse square law, and light equals 1 over d to the power of d squared thing. And an increase in light intensity will alter the rate of the light-dependent reaction. So more light energy is available to excite more electrons. More electrons take part in photophosphorylation, so increased light intensity means that more ATP and more reduced NADP will be produced. These are both used in the light-independent stage, the Calvin cycle, as sources of hydrogen energy to reduce glycerate phosphate, GP, to triose phosphate, TP. ATP is also used to phosphorylate 5 out of every 6 molecules of TP to regenerate RU-BP. Now, if there's no or very little light available, the light-dependent stage will stop. This will stop the light-independent stage as it needs the product of the light-dependent stage. That means that glycerate phosphate can't be changed to triose phosphate, so glycerate phosphate will accumulate and levels of TP will fall. This will lower the amount of RU-BP, reducing the fixation of carbon dioxide and the formation of more glycerate phosphate. That's light intensity. Now onto carbon dioxide concentration. Well, an increase in carbon dioxide concentration leads to an increase in carbon dioxide fixation in the Calvin cycle. This provides that light intensity isn't limiting. More carbon dioxide fixation leads to more molecules of GP, some of which might be converted to fatty acids or amino acids. And that means that there'll be more molecules of TP. These can be converted to hexose, sugars, disaccharides, polysaccharides, glycerol, and more regeneration of RU-BP. However, the number of stomata that open to allow gaseous exchange leads to increased transpiration, and this may lead the plant to start wilting if its water uptake from the soil can't exceed water loss by transpiration. This in turn leads to a stress response, and following a release of plant growth regulator, stomata close. This reduces carbon dioxide uptake and reduces the rate of photosynthesis. If carbon dioxide concentration is reduced between 0.01%, then RU-BP, the carbon dioxide acceptor, will accumulate, and as a result, levels of GP and subsequently of TP will fall. Finally, temperature. Increasing the temperature will have little effect upon the rate of light-dependent reaction, as apart from photolysis of water, it's not dependent upon enzymes. However, it will alter the rate of the light-independent reaction, as that of a series of biochemical steps, each catalysed by a specific enzyme. Increasing temperature will, at first, increase the rate of photosynthesis. However, as temperatures rise above 25 degrees, the oxygenase activity of Rubisco increases more than its carboxylase activity increases. This means that photorespiration exceeds photosynthesis. As a result, ATP and reduced NADP from the light-dependent reaction are dissipated and wasted, and this reduces the overall rate of photosynthesis. If you have very high temperatures, this might also damage proteins involved in photosynthesis. Increased temperatures cause an increase in water loss from leaves by transpiration. This could lead to the closure of stomata and subsequently reduces the rate of photosynthesis. Now, a good examiner tip is that we might get questions that expect us to interpret graphs from what happens to relative concentrations of GP, TP or RU-BP when factors changed. If you make a quick sketch of the Calvin cycle, you can easily explain why levels of chemicals rise and fall, because it's a lot simpler when you're looking at the Calvin cycle, because if they're like, oh, what happens to levels of this? You can be like, oh, well, if this doesn't happen, then this can't happen, then this can't happen. Oh, that means that this can't happen. Okay, I hope you understood what I just tried to mean there. Basically, if you have it all written out, you can see how things will affect different things, if you get what I mean. So like, yeah, so if you just sketch out the Calvin cycle, if you don't have it, there you go. So if you just basically sketch that out before you start to answer the question, it will help, because you'll be able to understand it better. So learn the Calvin cycle, that'll be good. Okay, that was quite a quick video actually, that's quite good. You've done approximately 5 minutes of revision there, so now you're allowed to go and have a half-an-hour break. That's definitely not what I do, ever. Alright, good luck in your exam, whenever you have it, and yeah, good luck.